Hello friends, welcome to the lecture series on botany. Today we are discussing Vaucheria, its important features and life history. Vaucheria is a single genus of family Vaucheriaceae, phylum Xanthophyta, the later commonly known as yellow-green algae. Let us start with the important features of Vaucheria. Vaucheria presently comprise of about 276 species which occur abundantly in the temperate regions of the world. Majority of these are found in terrestrial and freshwater ecosystems, as a few species are marine, like Vaucheria pyloboloides. The terrestrial species form extensive yellowish or deep green dense covering or mat on wet soil and in flower pots in greenhouses, commonly known as water felt. Two widely distributed species are Vaucheria sessilis and Vaucheria geminata. Vaucheria sessilis occurs both on land and in water, while Vaucheria uncinata is aquatic and Vaucheria amphibia is amphibious. Thallus structure. The thallus is composed of yellowish-green, cylindrical, coarse filaments branched at irregular intervals. In terrestrial species, the thallus is attached to the substratum by small tufts of colorless rhizoids or a lobed hapteron. The aerial filaments possess monopodial branching and apical growth. As there is no septa or cross walls, the protoplasm is continuous along its entire length. Septa occurs only during injury and formation of reproductive structures. The filament is weak, thin and inelastic. It consists of an inner layer of cellulose and outer layer of pectic substances. Inside of the cell wall, there is a layer of cytoplasm containing cell organelles. The chromatophore contains chlorophyll A, chlorophyll E, carotenoids. It lacks chlorophyll B. Pyrenoids are almost absent. The reserve food is oil or fat and gets stored within the cytoplasm in the form of countless droplets. In Vaucheria, cytokinesis is not followed by mitosis, so the thallus has xenocytic nature. In Vaucheria, plastids can change their orientation in response to change in light levels. In Vaucheria, chloroplasts, along with small vesicles and mitochondria, move with the streaming of cytoplasm along with longitudinal axis of the filament in dark. Cytoplasmic fibrils have been reported in the family of Vaucheria littoria. The photoreceptors of light-oriented movement of chloroplast are present in the cytoplasm rather than in the chloroplast. Vaucheria is an acellular organism. It is wrong to call Vaucheria as a unicellular organism. It possesses all the features of multicellular organism. Also, in unicellular forms, growth consists of an increase in size of the entire cell. However, in Vaucheria, growth is apical. Therefore, it is appropriate to call Vaucheria in general as an acellular xenocytic organism rather than a unicellular or multicellular form. Now, we have the second part of our lecture, that is life cycle of Vaucheria. Vaucheria reproduces by three methods: vegetative, asexual and sexual. Let us start with the vegetative reproduction. It takes place through fragmentation. In this method, the thallus breaks up externally into short segments which ultimately form new individuals. In Vaucheria, asexual reproduction takes place by different modes depending upon the habitat in which the alga lives. In aquatic species, it occurs by formation of zoospores. Terrestrial species form zoospores only when flooded. Zoospore formation. It is the commonest method of asexual reproduction in aquatic forms. Zoospores are large, multinucleate and multiflagellate structures. They are formed singly within elongated, club-shaped zoosporangia, which develop at the end of side branches. The zoosporangia gets swollen into a club-shaped structure. The entire protoplast of the zoosporangium contracts to form an oval multinucleate mass, the incipient zoospores. The mature zoospore escape through a narrow aperture which is formed by the gelatinization of the wall of the distal end of the zoosporangium. Now the morphology of zoospores. The multinucleate and multiflagellate zoospores are peculiar to Vaucheria. All other genera related to it produce small, biflagellate, uninucleate zoospores developed in large numbers in each zoosporangium. The zoospores are of compound structure formed as a result of the failure of protoplast within the zoosporangium to divide into uninucleate, biflagellate zoospores. Thus, it may be more appropriate to term it as a synzoospore, a view supported by two facts. Number one, presence of the central vacuole of the parent zoosporangium, and number second, paired disposition of the flagella opposite the nuclei. Now the germination of zoospores. After a period of 5 to 15 minutes of sluggish movement, the zoospores come to rest, flagella becomes motionless and then vanish completely. The zoospores round off and becomes covered with a thin cellulose wall. At this stage, the chromatophores move outwards and nuclei move inwards. The zoospore then elongates in one or two opposite directions in the form of tubular outgrowths. One of these undergo branching to form the colorless lobed holdfast, while the other continues to grow indefinitely to produce the yellowish-green tubular filaments. Another method of asexual reproduction is formation of epilenospores. These are non-motile asexual spores produced by the terrestrial species. The aquatic species produce them only when the algae is exposed to drought or transferred from light to darkness or from running to still water. The epilenospores develop at the ends of short laterals or terminal branches in epilenosporangia. The terminal epilenosporangia is cut off by septum from the branch. The protoplast of the epilenosporangium is converted into a single rounded thin-walled epilenospore which is liberated by the irregular rupture of the sporangial wall. The single epilenospore produced within the epilenosporangia is set free through the apical pore formed by the dissolution of the sporangial membrane. After liberation, epilenospore germinates to give rise to a new thallus. Now, another method of asexual reproduction is formation of akinets. In some aquatic and terrestrial species, when exposed to greater desiccation or low temperature, the branched filaments divide into a row of short segments by thick gelatinous cross walls. These resting multinucleate thick-walled segments are known as the cysts or hypnospores, or they are also called as akinets. The cyst in a chain may remain connected by the parent membrane of the filament, which appears like another alga, Vaucheria. Thus, this stage of Vaucheria is also called Vaucheria stage. Onset of favorable conditions for growth, the echinoids directly grow into a new individual. Now, we have sexual reproduction of Vaucheria. All species of Vaucheria reproduce sexually. It is oogame style. Most of the species are monoecious, that is homothelic. They are exclusively freshwater or terrestrial forms. Species such as Vaucheria dichotoma, which are marine, is dioecious, that is heterothelic. The male and female gametes differ greatly in size, form and structure. They are produced in the distinct and specialized sex organs. The male sex organ is called antheridium and female sex organ is oogonium. Position of sex organs. In the monoecious or homothelic species, the antheridia and oogonia usually occur close to one another at intervals along the same filament arising as a lateral outgrowths. There is a great variation in the arrangement of sex organs in the different species or even different individuals of the same species. Let's start with the structure of sex organs. Antheridia. In Vaucheria, the major antheridium is a cylindrical tubular structure strongly covered like a horn. In some cases, it is a straight. The young antheridium contains cytoplasm, nuclei and chloroplast. At maturity, it contains numerous male gametes or sperms, which in some species are liberated through a single aperture and in others through several apertures. The liberated sperm is an extremely minute oval spindle-shaped or pear-shaped colorless structure with two laterally inserted and opposite flagella of unequal length. The short one points forwards and long one points backwards. Now, oogonium. The oogonium is a spherical or wide structure which usually appears sessile or subsessile. It is separated from the supporting filament by a cross wall at its base and develops a short rounded beak at the tip towards maturity. The mature oogonium contains a single large nucleus located in the center and numerous chromatophores. Reserve food is stored in the form of oil droplets. The protoplasmic contents round off to form a single large egg cell or ovum. Now, fertilization. Both the sex organs open almost simultaneously, the oogonium a little before the antheridium. The antheridia and oogonia situated close together may dehisce almost same time, or either of them may open from a few minutes to 2 hours before the other dehisces. At the tip of the beak of the oogonium is formed a pore where from a colorless cytoplasm oozes out through the apical aperture. At this stage, the chromatophores and oil droplets move to the center of the ovum, which is surrounded by a colorless layer of lining cytoplasm. Several sperms emitted from the apical pore in the wall of antheridium gather around it. The small male nucleus lies near the female nucleus and increases in size until it has swollen to nearly the same volume before it fuses with the female nucleus. A membrane is formed across the oogonial aperture after fertilization. The zygote becomes oospore after secreting several layers thick wall around itself. When the original wall gets decayed, the oospore gets liberated. It then undergoes a period of rest. The resting zygote contains a number of reddish or brownish bodies. Now the germination of oospore. After the dormant period, the oospore germinates directly into a new filament without the formation of meiospores. It is held that reduction occurs in the first nuclear division in the germinating oospore. In that case, the thallus is haploid. Now, according to Mundi, 1929, Vaucheria thallus is diploid and meiosis occurs at the time of gamete formation. The thick oospore wall ruptures. Through the split emerges colorless germ tube or as a lateral outgrowth of the germ tube which forms the aerial system. Now we have taxonomic position of Vaucheria. The name Vaucheria was first given by D. Kendall in 1805. Strain 1948 made a critical study of the pigment of a few species of Vaucheria and came to the conclusion that they possess pigments characteristic of xanthophyceae. On the basis of pigment constitution of Vaucheria, Smith 1950 transferred this genus to class xanthophyceae. Many allergologists at present favor inclusion of family Vaucheriaceae with single genus Vaucheria in the class xanthophyceae. The arguments advanced in favor of this are: number one, the absence of native cellulose in the filament wall. Number second, absence of chlorophyll B and presence of chlorophyll E in the zoospores of Vaucheria, a pigment characteristic of class xanthophyceae. Presence of carotenoid pigments in excess of chlorophylls. Lack of two xanthophylls, siphonein and siphoxanthine, which are of universal occurrence in the siphonales. Absence of pyrenoids in the chromatophores. Oil and fat rather than starch is the principal food reserve. Presence of oppositely directed unequal flagella, a feature typical of motile cells of all xanthophyceae. On the basis of these features, the inclusion of Vaucheria in the class xanthophyceae has practically become a settled fact. Thus, according to the latest view, the systematic position of Vaucheria is as follows. Division: Xanthophyta, class Xanthophyceae, order Heterosiphoneales, family Vaucheriaceae, genus Vaucheria. Now, before we conclude, let's have a look on affinities of Vaucheria. In siphonaceous cynocytic acellular organization of the thallus, photosynthetic pigments, discoid chloroplast, cell wall composition, flagella morphology of motile cells, and principal food reserve, Vaucheria indicates relationship with other members of the xanthophyceae. The multinucleate, accepted thallus, and oogame sexual reproduction are the features in which Vaucheria resembles the green algae. Now, in cynocytic nature of the thallus, chemical composition of the cell wall, and oogame in the sexual phase, Vaucheria exhibits a striking resemblance with the oomycetes. The discovery of tinsel and whiplash type of flagella in the motile stage of Vaucheria is indicative of the mycomycetes having been derived from Vaucheria like ancestors. So friends, I hope that you might have understood the important features and life history of Vaucheria. See you with a new topic. Till then, goodbye.